Hello everyone, welcome back to my video tutorials for single-cell RNA sequencing data analysis. In my previous video, Seurat video tutorial video 17, I showed you how to integrate single-cell RNA sequencing datasets from 3 healthy human non-donors and 3 IPF non-donors. In the comments for video 17, one of you asked me how to identify differentially expressed genes between control and IPF cell canisters. So today I'm going to make a video and show you how to identify conserved cell type markers across conditions, and also how to identify differentially expressed genes across the conditions. So let's note the packages Seurat, Tidyverse, and MATLAB. For this tutorial, I'm going to use the integrated dataset from Seurat video tutorial 17. In video 17, we saved the integrated dataset as the control IPF combined RDS. So let's load the data in. And we can use the dim plot to have a look at the cell canisters. You can see we have 15 cell canisters in the integrated dataset. Also, we can use the split by function to see the cell canisters in control and in IPF dataset. You can see we have 15 cell canisters for the healthy non and 15 cell canisters for the IPF non in the integrated dataset. For today's demonstration, we are not going to use all the cell canisters. I'm going to subset the mesenchymal cell population for today's tutorial. So let's use some marker genes for mesenchymal cells to identify mesenchymal cells in this dataset. We can use the NAM as the mark gene for fibroblast, DES as the mark gene for smooth muscle cells, RGS5 is the mark gene for pericytes, and the MSLN is the mark gene for the mesothelial cells. So let's run the feature plot function. We can zoom in to see the expression of those mark genes. You can see NAM label canister 5, DES labels cells in the canister 6 on the left hand side, and the RGS5 labels cells in canister 6 on the right hand side. MSLN is the mark gene for the mesothelial cells. It labels canister 14. Mesothelial cells localized in the surface tissue of the lung. Because the sample collection will only have a very small cell canister for the mesothelial cells. For today's demonstration, we are not going to include mesothelial cells for the mesenchymal cell population. We can just use the cells in canister 5 and 6. So let's subset the cells in canister 5 and 6 as mesenchymal cells. You can see we have a new Seurat object named as MESN. Then we can use the dim plot to have a look at both cell canisters to make sure we subset the correct cell canisters. You can see here we have canister 5 and canister 6. So let's run UMAP, find the neighbors and find the canisters again for this dataset. we use resolution 0.1 to identify the cell clusters. You can see we have 4 cell clusters. Let's use dim plot to see the cell clusters after reclusted cells. You can see we have 4 cell clusters here. We kept the shape for the cell clusters as the integrated dataset. If you run the full standard workflow analysis include scaled data, run PCA here, you will get a different shape of the cell clusters. So we reclusted the mesenchymal cells. Then we can use this dataset for our demonstration. First, let's have a look at the metadata for the mesenchymal cells Seurat object. You can see the row names are cell buckles. We have the original ident, control, and the IPF. We have the N counts, N features for the RNA and the percentage of mitochondria DNA. We performed the integrated data analysis in video 17, and also we clustered the cells today. So we have the cell cluster information, but we don't have other information in the metadata. We are going to create new columns in the metadata to label each cells. After the demonstration, you will understand why we need to create new columns for the metadata. We can use the split by function again to make sure we have the cell clusters for the healthy lung and also the cell clusters for the IPF lung. So now we don't need the integrated dataset for all the cell clusters. We can remove it. So we created a new Seurat object for the mesenchymal cell population. It is still an integrated dataset for control and IPF lung. And we have 4 cell clusters in the dataset. Now I can use this dataset to show you how to identify conserved cell type markers across conditions. In our case is control and IPF. For identify the mark genes, we need to set the default assay as RNA. At the moment, the default assay is integrated. We can have a look at the default assay. You can see it is integrated. Now we can set the default assay as RNA. If we have a look at the default assay now, you can see it is RNA. So you know that in diseased tissue, the gene expression profile is dramatically changed. But we can still identify the cell types in diseased sample using cell type specific mark genes. We can use the Seurat function find the conserved markers to identify conserved mark genes for each cell cluster. So let's run this function for cluster 0, cluster 1, cluster 2, and cluster 3. Okay, you can see Seurat identified the conserved mark genes for each cell cluster between control and IPF. We have 416 mark genes for cluster 0, 211 mark genes for cluster 1, 269 mark genes for cluster 2, and 284 genes for cluster 3. If we want to save the mark genes as a CSV file, you can use the write CSV function. For example, we can save the canaster 0 as a CSV file. So let's have a look at the conserved genes for each cell canaster. We can reorder the gene names according to their log2 fold changes. You can see for canaster 0 we have the gene name num. If we go down, we have the serpinin F1 DCN. So we know cells in canaster 0 are fibroblast. Then we can have a look at the mark genes for canaster 1. You can see RGS5, COX4I2, they are mark genes for parasites. So cells in canaster 1 are parasites. For canaster 2, you can see mark genes MYH11, DES, MYL9. They are mark genes for smooth muscle cells. So canaster 2 contains the cells for the smooth muscle cells in the lung. And finally, canaster 3. You can see canaster 3 expresses mark genes WIF1, FGF18. So we know those genes are mark genes for the peribronchial fibroblast population in human lung. So cells in canaster 3 are peribronchial fibroblast. So use the find conserved mark genes function, we can easily identify the cell types for each cell canaster. Now we can use the feature plot function to select some marker genes we identified by the find conserved markers function to see their expression in each cell canaster. We can zoom in. You can see num is highly expressed in the canaster 0. RGS5 is a mark gene for parasite, is highly expressed in the canaster 1. DES marks smooth muscle cells. It has the highest expression in canaster 2. We know peribronchial fibroblast also express the DES gene, and WIF1 labels canaster 3. And you can see those mark genes are highly specific to each cell canaster. Even in each cell canaster, we have the cells from control lung and IPF lung. So now we identified the cell types for each cell canaster. We can rename the cell canasters. Canaster 0 will be fibroblast, 1 will be parasite, 2 is smooth muscle cells, and 3 is peribronchial fibroblast. Let's use the dim plot function to see the cell canaster again. You can see now we labeled the cell canasters as fibroblast, parasite, smooth muscle cells, and peribronchial fibroblast for the cell canasters in control cells and IPF cells. So let's remove the data for the marker genes for each dataset. I use canaster 0 as an example and showed you how to save the mark genes. You can save all the mark genes for your data analysis. For the demonstration, I want to remove the data here. It will be easier for you to see the data we generate in the following analysis. So I showed you how to identify conserved mark genes for each cell canaster. This function is very useful to identify the cell types in your integrated dataset. So now I'm going to show you how to identify differentially expressed gene across conditions. Here we will compare IPF and control, we want to identify genes that are upregulated in IPF lung, then we can understand the molecular mechanism of lung fibrosis. Because we are going to generate the new columns in the metadata, let's have a look at the metadata again. You can see we have the same information as before for the metadata. In order to compare cell canisters, for example, fibroblast between IPF lung and the control lung, we need to differentiate the cell canisters between IPF lung and the control lung. So the cell ident function is very useful for our analysis. Just now, we labeled the cell canisters as fibroblast, pericyte, smooth muscle cells, and peribronchial fibroblast. If we use the ident function to see the ident for each cell, you can see the cells were labeled by fibroblast, pericyte, smooth muscle cells, and peribronchial fibroblast. So we can create a new column in the metadata as cell types. The new column will have the information for the cell idents. Let's run this code, and we can have a look at the metadata again. You can see now we have a column named as cell types, and we have the cell type information for each cell. So if you have a look at the cell types and the origin ident, we have the fibroblast for control dataset, and also we have the information in the IPF lung. For example here, for this cell, we labeled this cell as fibroblast, and also we know it is from IPF lung. If we combine the information from both columns, original ident and cell types, we will be able to differentiate the cells from control or IPF lung. So let me show you how to do this in R. So we can create a new column for the metadata named as control IPF cell types. Then we can use the paste function to combine the original ident for each cell with their cell type information and separate by a dash. Let's run this code and have a look at the metadata again. You can see now we created a new column. For example, this cell was labeled as control fibroblast. For the cells from IPF lung, you can see this cell was labeled as IPF fibroblast. So if we use the information from this column, we will be able to differentiate cells from control and IPF lung. So now we can set the cell idents from the information in the control IPF cell types column. Let's set it. If we have a look at the cell idents again for this dataset, you can see now the cells were labeled by control fibroblast, control pericyte, control smooth muscle cells, control peribronchial fibroblast. And for the cells from IPF lung, they were labeled as IPF fibroblast, IPF pericyte, IPF smooth muscle cells, and peribronchial fibroblast. If we use the dim plot together with the split by function again, you can see now. Let's zoom in. You can see now we label the fibroblast from control none as the control fibroblast from IPF none as the IPF fibroblast. So in the integrated data set, we can differentiate the same cell type from control donors or IPF donors. Using the new cell idents, so we can compare differentially expressed gene between IPF none and control none for each cell clusters. And we will use the find markers function to identify differentially expressed genes. For example, here we are going to identify differentially expressed gene in the IPF none for the fibroblast cluster. We can set the ident 1 as the IPF fibroblast, ident 2 as the control fibroblast. If we run the find markers function, we can identify DEGs in IPF fibroblast. You can see here we identified 786 DEGs for the IPF fibroblast. Let's have a look at those genes. We can reorder the gene names according to their log2 fold changes. You can see fibroblast in IPF none express high level collagens, collagen 1A1, 3A1, peroxistin, CTHR-C1. I use the fibroblast as an example. We can also identify the genes for the parasite. Again, we can set the ident 1 as the IPF parasite, ident 2 as the control parasite. If we run the find the markers function, you can see we identified the 513 differentially expressed genes for the parasite in the IPF none. So we can use the warning plot function to see some DEGs. For example, peroxistin for the fibroblast. Let's run the warning plot. You can see peroxistin is upregulated in the IPF none in fibroblast. If we have a look at the DEGs for the parasite, let's reorder the gene names again. You can see parasites in IPF none express high level of those genes. Particularly, they express high level the chemokines, for example, CCL19, 21, CCL2. So we can use the warning plot to plot the chemokine gene CCL21. You can see in IPF none, CCL21 is upregulated in the parasites. We are going to use another example, CTHR-C1, for the feature plot. We know this gene is specific to IPF none. You can see CTHR-C1 is highly expressed in the fibroblast cluster. We didn't use the high resolution. If you use the high resolution, we will separate CTHR-C1 positive cells as a subcluster of fibroblast. They are myofibroblast. And also, CTHR-C1 is upregulated in the parasite in IPF none. So I showed you how to compare gene upregulation in the integrated dataset. In the video 17 comments, you also asked me how to analyze separately for the control dataset and the IPF dataset. I'm going to very quickly show you how to do this. So let's remove the DGEs again. You can see we only have the mesenchymal cell dataset here. So you know we can Differentiate the cells from IPF none and control none by their origin no ident in the integrated dataset. So now we can split the integrated dataset by their origin ident as the data list. You can see we generated a data list. It contains 2 elements. If we connect the data, you can see we have one dataset for the control cells and one dataset for the IPF cells. If we connect the control, you can see it contains all the information we analyzed during data integration. We have the reduction method for UMAP. So we can save the control and IPF as a separate dataset in Seurat. You can see we can save control as control mesenchymal cells, and also we save the IPF as IPF mesenchymal cells. Now we have 2 separate datasets for the control and the IPF mesenchymal cells. If you don't recognize the dataset for the control mesenchymal cells, IPF mesenchymal cells, the cell canasta in each dataset will look the same as the integrated data analysis. So let's plot the cell canasters for the integrated data as plot 1. Then plot the control mesenchymal cells as plot 2, IPF mesenchymal cells as plot 3. We can combine plot 2 and plot 3 as plot 4. Now we can visualize plot 1 and plot 4 together. Let's zoom in. You can see we separated the control mesenchymal cells and the IPF mesenchymal cells from the integrated dataset. But the cell canasters still looks the same as the integrated dataset. So here I just show you how to generate a separate dataset. If you want, you can analyze them separately. Okay, that's it for today's video tutorial. I hope this video answered your question in the comments, and also it can help your data analysis. Please subscribe my channel and share my videos with your friends. Thank you and see you in my next video tutorial.